Good morning, everybody. Awesome. Welcome to Science on Saturday. We're so grateful that you're spending your Saturday morning with us. I'm Dr. Joanna Albala. I'm the manager of the Science Education Program at Lawrence Livermore National Laboratory. And I am so excited that you are here for this year's theme, Computing the Future, where we're showcasing our innovative work using the power of computing from molecular biology to space exploration. Without any further ado, let's welcome our presenters. Dan Faissol received a BS in Electrical Engineering from the University of California, Los Angeles, an MS in Economics, and a PhD in Industrial and Systems Engineering from the Georgia Institute of Technology. He is a distinguished member of technical staff and a principal investigator for the Generative, Unconstrained, Intelligent Drug Engineering or GUIDE program at the laboratory. He's joined today by Erin McKay, who received her BS in biology with an emphasis in plant biology and her science teaching credential from UC Davis, and she is a biology teacher at Tracy High. She is also a faculty scholar in the science education program at the lab. Today's talk is entitled High-Tech Virus Fighters, AI, Supercomputers, and Biology on the Front Lines. Now let's get started.
All right, thank you for having me. Thanks for coming. I am super excited to talk about what we're doing at Lawrence Livermore to fight viruses. Super pumped because we're doing awesome work. It's very, very cool stuff. And we have some exciting stuff to talk about today. So first, a little bit about myself. So I actually am from the Bay Area. I went to middle school and high school not far from here in Fremont, California. It's about 20 minutes from here, half an hour, I don't know. And from there, I went to college and studied engineering, and then got a PhD. And then from there, I went straight to Lawrence Livermore. I've worked there ever since. Love working there. We work on really, really cool problems that can change the world. So love working there. I have 2 little kids, a 5-year-old and an 8-year-old. And my wife is actually a teacher at Livermore High School. She teaches engineering in the Green Engineering Academy. So we're very local. What I do for fun on my free time when I'm not playing with my kids is I go e-foiling. This is a picture of me e-foiling. E-foiling is super cool. It's basically a mini surfboard with a motor underwater that just propels you really fast, and you get to like fly and hover above the water. Super fun. Here's me doing it in the Bay Area. I was hoping to talk the whole hour about this, but Joanna said 1 slide is all I get. So we have to move on. All right. But the only thing cooler than e-foiling is what I'm going to talk about today. So what we're going to actually learn about today is how viruses infect our bodies, how our immune system can fight viruses with antibodies, and how we can now design antibodies using AI and supercomputers. That's what my talk is about today. I'll walk you through each of these steps, and I'll give you a really cool case study of what we did a few years ago. Okay, so first thing I'll mention is actually at the beginning of the COVID pandemic, I actually gave a Science on Saturday talk on the work that we had been doing for COVID. And this is the same slide I showed then a few years ago, where I'm still blown away by this, right? The new virus emerges out of nowhere. It didn't exist before. And then all of a sudden, it just exists. And in a couple of months, the whole world is infected. Every piece of the globe, COVID reached. And in a few months after it started, 1 in 7 people in the US had been infected. I'm still blown away by that. And this is just one example. There have been many other pandemics throughout human history. And this is just one virus. There's lots of viruses. And they're all very, very different. Some, like COVID, can go through the whole globe very, very quickly. Others are more localized, and they have outbreaks in parts of the world. And they can have very high mortality rates, much higher than COVID and flu, but they're localized. And then on the other end of the spectrum, these ones that hit the whole globe, and everywhere in between. So viruses have been around pretty much as long as humans have, and we've coexisted with them. And there's huge numbers of viruses and different types. So what we're talking about today is how we can fight viruses. Okay, vaccines and drugs are 2 key ways to fight viruses. We know about vaccines, we know about drugs. Therapeutic antibodies are a major type of drug that can help with viruses. I'll talk about those today. They take a very, very long time to develop, both vaccines and these therapeutic drugs. And this talk is about how we can accelerate that and do it very quickly. Okay, so our challenge to fight viruses is how can we go super fast? Instead of taking a whole year or more, can we design them or re-engineer them very, very quickly? But it's not just about speed. We have to be able to do it and make these antibodies very safe. If we're going to give them to people, they can't have other effects on the body. They have to be very safe to people, and they have to be effective. So fast, effective, and safe. Those are hard challenges. Okay, so a little bit about viruses first. How do viruses work? This is the example of the SARS-CoV-2 virus, the virus behind the COVID-19 pandemic. So viruses, this virus in particular, many, many viruses like it, actually have this secret weapon on the surface of it. This red part is called the spike glycoprotein. This protein is the virus's secret weapon to attach to one of our cells in our body and invade it. This is how it works. Here's a cartoon of the virus with the same spike glycoproteins in red on the outside. When the virus enters your body, it'll float around and it'll bump into this cell receptor on a human cell. Now that spike glycoprotein on the virus, that red part, it is perfectly shaped to match like a lock and key to that part of our cell, the cell receptor. And so it has evolved that shape to be able to do that. So it binds right there perfectly. And what it does is then it's able to inject its DNA, its RNA actually, into our cell, which is the key to how it all happens. So let me pause there for a second and explain why this is important and how this works in general. It's called the central dogma in biology. You've probably heard of DNA. DNA, double-stranded helix, it encodes for everything in our bodies, right? It's like and in this case, it's like a cookbook for making proteins. So when your body needs a protein, it'll take the DNA, it'll extract a piece of it, and it's transcribed into RNA. RNA is single-stranded, that gets translated into a protein. These proteins are then physical things that do things in our body. It can help build muscle, it can digest food, or it can fight viruses. All sorts of activities in our bodies require these proteins, and that's how our cells make them. They make them from DNA to RNA to proteins. And these proteins are encoded by those amino acids. So Erin will talk more about that and do a demo about how that works later. But for now, know that this virus, it doesn't even have DNA. It's got just RNA inside of it, and it's using that red spike glycoprotein to attach to the cell, and then now it's able to insert its RNA into our cell. And this is where it gets really, really sneaky. It uses our cell's machinery to replicate itself. So our cell doesn't know that this RNA is from the virus. It does what it does. It sees RNA, it translates that into protein. It turns out in this case, this is the virus that it's replicating. And so we get tons more and more virus, and that's how we get sick. So the virus has tricked us into replicating itself, and we get more and more sick. Super uncool, super uncool and sneaky. But it turns out our immune systems can be very, very sneaky and fight back against this. So one way how we stop viral infections, we know, right? We cover our mouth when we cough, we wash our hands. But another way is antibodies. Our immune systems have developed these antibodies to fight these viral infections. So this is how it works. This is a 3D picture or a 3D model of an antibody. They're very, very complicated proteins that our bodies make. Here's one in more globular form to show. And I'll rotate it so you can see how complicated the 3D shape is. This is just sort of the backbone, if you will, of the antibody. There's actually lots more complication involved in it with amino acids that give it a specific chemistry. But this is the shape of the antibody, and its shape is what determines what viruses it can bind to and defend against. So here's how it works. Here's a cartoon of the antibody. And remember I mentioned the RNA and the DNA and the amino acids and the protein? This is the amino acid sequence encoded that codes for the protein. So the shape of the antibody is determined by this code here that I'm showing, this sequence of letters, right? It's an alphabet code of letters, very long and complicated. That code is what makes the shape of this antibody. So for a given antibody that fights against COVID, there'll be one code. For an antibody that fights against flu, it'll be a different code. Okay, so here's how it works. The antibody will then bind to that spike glycoprotein. This is one way antibodies work. It's a very powerful way in which they work. And it's like a lock and key. So this antibody only binds this virus. It was developed by your immune system to bind this virus and stop it from replicating. And we make many, many of these antibodies in our bodies to fight different kinds of infections that we get throughout our lives. So here's how it works. We have the antibodies floating in our body. They bind to that secret weapon of the virus, the spike glycoprotein. And when they bind, they stay there, and they prevent it from binding the part of our cell where the virus attaches. And then when it does that, it prevents the virus from doing its thing, and it doesn't replicate anymore. So that prevents us from getting sick. It stops the virus in its track completely. So antibodies are awesome. They do that. Okay, so making antibodies can save lives and prevent people from getting sick. We can now make antibodies and give them to people as drugs, just like other drugs that we know about. So antibodies are made in a laboratory and they're given to people. And these are especially important in cases where people's immune systems don't work so well. And there's lots of people like that, including newborns, elderly, people that are immune compromised. Their immune systems don't make the antibodies that do what I just showed. So for those people especially, we can make these antibodies and give it to them to protect them from these viruses. Okay, so I'll give you a case study of an antibody that was saving lives in the real world against SARS-CoV-2, against COVID. During about a year into the pandemic, these antibodies were developed and they were provided to people and they were working. Then, as you may know, the virus mutates throughout its course. During the pandemic, the virus that first emerged, SARS-2, it evolved. And it specifically is trying to evolve to do various things. One of the things is to try to escape our antibody response, meaning it's trying to change the shape of its glycoprotein so that our antibodies don't bind to it anymore. Okay, it does that with the same RNA that it has. That RNA it's in the middle of the virus, it encodes for the shape of that spike glycoprotein. And so it mutates that code. It changes that code so that it doesn't bind our antibodies anymore. And in this case, it was called Omicron. It changed its code, and it stopped all the antibodies that were around at the time from working anymore. That was a huge deal. It was a red alert. It was a red alert. In fact, we had been working on this approach to design antibodies. We still are working on it. We weren't done. And we got a call from the government saying, hey, Omicron happened. The antibodies that we have stopped working. They aren't working against SARS anymore. Can you guys help? Can you guys help part of this response that fixes this? This is like straight out of a movie. We got a call from the government saying, can you help? And I said, who's this? No, I didn't say that. I said, okay, we can do this. We can help. We can help. We're ready. We're ready. We're on this. We actually weren't ready. We were still developing our whole thing at the time, but they knew that. But we were going to go for it. We were going to go for it. So our job was to fix the antibodies or design a new one to compensate for this change that happened in the virus. So essentially, with the lock and key, how antibodies bind to their virus with a lock and key, basically what happened was the key, or the lock has now changed. And the original key, the antibody, didn't work to open it anymore. So we have to make a new key. We have to find a new key or design a new key. So this was called Omicron. Omicron was the mutated virus that caused all this to happen. Okay, so now the antibodies no longer bind the virus because it changed its shape, and then everything goes back to happening. Now we can get sick again, and we need to be able to fix the antibody or provide a new antibody so that it stops this whole cycle from happening. That was our challenge. That was our mission. We need a new key. It's very, very hard to find a new key, and it takes years. It could take more than a year. It typically takes more than a year And so we were asked, can you do it really, really fast? Can you do it in 3 weeks? That's what we went for. We went to try to design a new one in 3 weeks. Hadn't been done before. Never been done before. So we went for it. Okay, so what are we going to do? So our idea was, let's start with the original antibody. So the original antibody was effective against the original SARS-2, no longer effective against the mutated SARS-2. So maybe we start with an antibody that does work against the original SARS and change it a little bit just right so that it does bind against Omicron. In the same way that Omicron mutated, we can try to mutate the antibody to compensate. That means we have to basically decrypt this code. We have to understand how this code, this amino acid sequence, how that converts to the shape of the antibody, and therefore how that makes it bind or not bind the mutated virus, Omicron. That's super hard to do. I can't read this code. Basically, it's gibberish to us, right? What does this mean? And how do we know what to change to give the antibody the right shape? Okay, we went for it. How do we change this code to make antibodies bind against Omicron? So I say there's a hard problem, and this puts it into context a little bit. Right, so chance of getting struck by lightning, 1 in 14,000. Seems a little low, actually, but okay. Getting bit by a shark or attacked by a shark, 1 in 11 million. The lottery has like 300 million possibilities, possible different numbers. Your chance of winning that if you have one ticket is 1 in 300 million. The antibody design problem that I just described is a quintillion possibilities of antibodies. That's 18 zeros on the end of that. That's huge. We're not going to be able to get that with luck, right? We have to come up with a very, very smart way to change this antibody, to fix it, to make it bind Omicron. Okay, so that's what we've been getting ready to do. We've been working on that problem for a few years already. How do we do that? Well, we've been using supercomputers together with AI to do that. So, what I'm showing on the right is a simulation where we are modeling every single atom on the antibody and the spike protein of the virus where they come into contact to try to understand, is that shape and chemistry, are they compatible? Will it bind like a lock and key? Right here in the middle, where the blue and the orange are touching each other, that's where they bind each other. And that's the part that we have to pay extra special attention to change those amino acids there so that the antibody that doesn't bind gets the right shape and chemistry so that it does bind. Super, super hard. Remember, it's a 1 in quintillion set of possibilities. Okay, so we're using AI models together with these simulations in a loop that I'll show you in a second to try to predict what changes to the antibody would make it bind. So it's like finding a needle in a haystack or a key in a giant pile of keys. Okay. We actually designed 376 different antibodies because we couldn't be sure which one of them would actually work. We're just making predictions in a computer model. Right? We're just modeling all this and trying to make predictions and hoping that one of them works really well. So we didn't want to take just one shot on goal. We took 376. We designed that, and so for each one, there's a different code. There's a different amino acid sequence that encodes for a different shape of an antibody that may or may not work in real life to block the virus from entering our cell. Okay, so how does this work? On the left, I'm showing an optimization loop. This is all happening inside the computer using those simulations I showed you earlier, where we model every single atom and the interactions between the antibody and that spike glycoprotein, that weapon on the virus that enters our cell. And we simulate, we get an answer, we update our AI model, and we propose another change to the amino acid sequence, a new key. And then we simulate that, and we keep trying, we keep going. And we continue that cycle of trying new keys, introducing smart, clever, targeted mutations into the antibody to try to make it bind. And then after we do that for a while, we end up with a set that we think, okay, these we hope work. Or at least one in this set we hope works really well. Maybe more. And then on the right, we're showing we go to the laboratory. Now, normally when we do this, we can do a cycle in the laboratory as well. We can try a batch of 100, take those results, and go back to the computer and simulate more and try to propose more with our AI models. And we could do multiple iterations of that. In this case, we had one shot. We were doing the computer modeling, and then it went straight to the laboratory. We had one shot to get it right and to find an antibody that actually is effective and protective against Omicron. Okay, so I will hand it over to Erin to show you a demo of proteins.
Thank you, Dan. So he's talked about how they use the computers to help do the simulations, but then how do you bring that into the laboratory and actually turn that into real antibodies and test whether they actually function? And so that's going to require that we go back to something that we study in middle school and we study even deeper in high school. And if you choose to go into biology or molecular genetics, you get even deeper into. And that is, let's go back to DNA. So I'm sure all of you have heard of DNA. It's pretty famous. Doesn't do much, actually, but it stores the information on how to build the things that do all the actual work. So up at the top, we see the DNA double helix, and we get a closer view. Maybe you recognize the A, T, C's, and G's, and you might notice how A pairs with T, C pairs with G. That's our base pairing rule to make sure that we get two complementary strands. And so what we need to be able to do is be able to figure out how to build that DNA that's going to code so that we can actually build the actual proteins with the correct amino acid sequences so we can make those 300 and something potential antibodies and test to see which one can actually do the job properly. So how do we actually get that DNA to build a protein? We saw a little preview of that earlier, and that is we need to bring out some RNA polymerase enzymes. And not pictured here, but what is the end result? RNA polymerase enzyme is going to read the DNA, and from there, it's going to do base pairing again. But you might notice when you look at that RNA, there's no T there for base pairing. Instead, oh wait, what is it? It's a U, because RNA does not have a T, it has a U. So we go from T pairing with A pairing with U, and C pairing with G. And we get the most famous RNA sequence, AUG, which is the start codon, which codes for methionine, M. So anytime you saw an M in the previous sequence of alphabet soup next to those antibodies, that was a methionine amino acid. And so reading in sets of 3, we've got AUG codes for methionine, AUC codes for isoleucine, which would be I in the single letter code, and then we would have UCG codes for serine or S in the single letter code, and you just keep going and going and going until you hit a stop codon, and then we're done. And so that is how we actually need to go about building the proteins, the antibodies. You got to go backwards, and you got to build that DNA, and then let the RNA polymerase build the RNA, and then the ribosomes build the protein. And ultimately, we would get our lovely little antibody protein. But actually, it's a pretty big jump between the sequence of amino acids and actually a properly folded protein. So let's explore that a little bit more. So here we see just a ball and stick model that shows us the amino acids just with the little color coding. Now, there are actually 20 common amino acids that are used to build proteins. A small protein is about 100 amino acids, and they interact with each other and they interact with their environment. And the first thing that they do is the first type of folding, what we call the secondary structure. One thing we can get is alpha helices. They look like little curly cues. There's another type of folding that I'll point out when we get back to the computational model, which is called beta pleated sheets. And then from there, that based on very unique little characteristics of the different amino acids and the environment that they're in, you start to get a tertiary folding, this overall structure. And for some proteins, there's more than one amino acid chain, what we call a polypeptide chain. And so we get quaternary, or the 4th level structure. And then we build our actual functional protein that's going to do the work. And so here we have the protein that we're trying to build, an antibody protein. Now, we're not trying to just build one antibody protein. We got over 300 different antibodies that we need to build with slightly different variations. So we got over 300 different types of DNA variations, go through DNA to RNA, RNA to protein, let it fold properly. Now, if you see the arrows going ziggy-zaggy back and forth, that's the alternative to the alpha helix, that little curly Q. That's called the beta-pleated sheet. Okay, so let's dive a little bit deeper into how it is possible that we can get our proteins to fold. First, I'm going to set this over here so it's safe. As I said, there are over 20 different amino acids that are used to build proteins put together in the unique order, and the length that the protein is will then allow it to fold up into its functional shape. Structure leads to function. Change structure, change function. And we can classify all of the amino acids into two very simplified categories, which are hydrophobic and hydrophilic. So we don't even need to have a really in-depth understanding of chemistry to at least appreciate this basic idea of what can drive protein folding. Another important thing to know is that if it is a protein folding in the cell's cytoplasm, the fluid of the cell, most of the cell's cytoplasm is made of water. Oh wait, we're going to be talking about hydrophobic amino acids and hydrophilic amino acids floating around in water and folding based on their interactions. Hydrophobic, hydrophilic. Hydro means water. Phobic, how many of you have heard of phobic before? Yeah, that's where you're scared of it. You don't like it, okay? And then philic, it means to love it. So here we have some exaggerated water. I added some blue dye, okay? And then we've got, oh, 2 different bottles. Uh-oh, I did bad science. I did not do any labeling. That was actually intentional. So I'm going to take this one. I think this one should be our oil. So water inherently is hydrophilic. It loves itself. It likes to stick to itself. Okay? And then I'm going to take some oil, which is famously hydrophobic. There's even a saying, oil and water don't mix. And so let's pour it in. Okay? And we can see that there's globules, globules, globules of the oil, and then it starts to float because it's less dense than the water. And we're starting to get a very, very distinct line right between, and so the oil and the water are not mixing. The hydrophobic oil likes to hang out with itself. The hydrophilic water likes to hang out with itself. But let's just double check, you know, science. Don't just do something once. So now I have some water also, but I added some yellow dye to it. Oh, it's a good thing that's not the classroom. So I'm going to add this in, and let's see what happens. Again, the oil separates out, and we notice, let me just, it's too big to stir it. I did not bring a big enough stir stick. But now we can see the green down here. So the blue dye and the yellow dye, which are also hydrophilic, water-loving, are hanging out down with the water. We got a little bit of a mixy zone, and then we've got all the oil sitting up top. So oil, hydrophobic stuff, likes to hang out with other hydrophobic stuff. Water and other hydrophilic molecules like to hang out together. And so here we can see our basic understanding of hydrophobic and hydrophilic. Going back to our protein folding, amino acids. There's 20 of them. That leads to 20 amino acids. Some are hydrophilic, some are hydrophobic. And to help us understand that even more, I've got 14 lovely Tracy High students who are going to be our hydrophilic and hydrophobic amino acids, and they're going to come up onto stage. You might notice that they're wearing different color shirts, and when they come on up here, you'll see that they've got some lovely emojis on themselves. I wonder which one are the hydrophilic ones. Might they be the one with the heart eyes? And the hydrophobic ones, oh, those are scary. And then we do have color coding. We have blue is the hydrophobic, gray is the hydrophilic, and you might be like, wait a minute, water is blue, we think. Yeah, sorry, got the counts wrong for the shirt sizes, so it is what it is. So we are going to be doing a human model simulation of multiple variations. So we're going to start off with, let's build a hydrophobic protein. Now, it's going to be a very tiny protein. It's only going to be 4 amino acids long. So can our 4 hydrophobic amino acids come forward? Yep, got to do that translation. Form those peptide bonds. They're just fancy words for covalent bonds. Okay, I'm going to hit run on the simulation. Run. Oh, they're in the cytoplasm which is watery. They do not like the water. They're trying to hide from the water, reduce the surface area, but it's a real environment, so they've got some kinetic energy going on, and they're just going to continue to bounce around there. Now let's take some hydrophilic amino acids. Come on forward. They've already joined up. Okay, so they are hydrophilic and they're in water, so they like the water, they like each other, they're just happy to bounce around. And so here we go. And now go ahead and take a step back. But most proteins aren't just hydrophobic or hydrophilic. They're a mixture of hydrophobic and hydrophilic amino acids. And so we're going to build a slightly more complex amino acid chain for our protein. But I think we need one more hydrophobic, so I'm going to steal one. Thank you. And I need one more hydrophilic, so I'm going to steal one. Okay, so now we've got a little bit of a mix-up going on here. Take a moment in your head. Try and imagine what's going to happen when they do this human modeling. So let's go ahead and step forward, form your peptide bonds. Okay. Run simulation. You're in water. What's going to happen? And remember, you're kinetic, so move around. There we go. Okay, so that's one possibility, but we got some very simple rules, so that's not the only way they could potentially fold up. Let's go ahead, reset simulation. Okay, let's see. Now imagine, what's another way they might potentially fold up just using these very simple hydrophobic-hydrophilic interaction rules? Come on forward, run simulation 2. Okay, there we go. And come on, you're in some fluid. Move it. Okay, there we go. Now, go ahead and we can see this is just with using a couple of very simple rules. And then this is actually something my freshman did yesterday in class with pipe cleaners and beads, was trying to simulate protein folding. Now, in the modern computational models, there's so much more detail into which amino acid likes which amino acid, which one doesn't, and running so many more simulations. But you can just see here what goes into protein folding and how it ties in with the fundamentals of what you study in middle school and high school of molecular genetics. DNA codes for RNA, RNA codes for protein, but a protein can't do its job till it's folded up properly. Thank you very much, Tracy High Biotech. Okay, and back over to you.
All right, thank you for that. That was awesome. So that was critical because you saw how complicated it was and all the movement happening and hydrophobic, hydrophilic for a protein just to fold, right? But we need these proteins to fold to make the drugs. Our bodies are awesome at doing this already. They're making proteins all the time, different kinds of proteins, complicated proteins. We can today make these proteins in a laboratory and give them as drugs, but that's very difficult to do. We have to set up the exactly perfect conditions and be very, very meticulous about doing it so that we get the protein to fold in the way that you just saw up on stage. It's very, very easy to do one little thing wrong and the protein completely misfolds. We don't get enough protein, therefore we don't have the antibody drug to give to a patient. So I'll talk about how that happens in the laboratory at Lawrence Livermore and with our partners. But first, let's do a quick review where we're at. Okay, so we had a red alert. There was a virus, a new virus, SARS-2, COVID-19 pandemic, infecting people all over the world. A year in, there was an antibody available to give to patients whose immune systems aren't fighting the virus. It's being used, it's saving lives. Omicron happened. What happened? The virus mutated. It escaped these antibodies. It deliberately mutated the shape of its spike glycoprotein on the surface so that the antibodies would no longer bind it. Red alert, government calls us. I say, who's this? Just kidding. No, we'll do this. We'll help you out. All right, we work on it. We spend 3 weeks cranking on our AI, supercomputers, simulating every single atom where the antibody binds that spike glycoprotein, and we come up with 376 possibilities that we think might bind the spike glycoprotein and therefore prevent the virus from entering the human cell. Okay, that's where we left off. But we got to make it in the laboratory and test that this actually works. Our computer models say, we think one of these will work. but we don't know. Okay, so we're going to make protein just like we showed on stage. There's 3 major steps. The 3 steps are, 1, the DNA part. We have to make the DNA. We have to assemble the bits of DNA and clone them and get them ready to put into a cell. So that's a long, that's called molecular biology. That's a complicated process to assemble these tiny little fragments into the right code, because that code has to be exact. It has to be exact. If we mess up that code a little bit, it won't code for the right protein. It probably won't even fold into a protein, or if it does, it'll be the wrong protein. It won't work. So we need an exact code there specified. Once we have that perfect, we can now put that into a cell, just like the virus hijacks a cell to make copies of itself. We can put this DNA into a cell that produces the protein we want, in this case, an antibody, right? The drug that we want to give patients to help them. Okay, that's also a very delicate process. We have to put it in exactly the right conditions and to allow the protein to be produced stably. Then the third step is we have to actually take that antibody that we've produced and test it to see, does it actually bind Omicron? Does it bind Omicron the way we want it to bind? And if it does bind, there's a second test that we do called neutralization. That means, does this antibody bind and bind well enough and stay bound long enough to prevent the virus from entering the cell? Like in that, when I showed you earlier, right? It binds there, it blocks the virus from using its spike glycoprotein to enter the cell. That's what we want. We have to test that. Okay, those are the 3 steps. So, at the time, we were making antibodies one by one. Each of those 376 would have had to be made one by hand in flasks and pipettes, putting liquids into flasks. We were at the time working with a partner, one of the world leaders in antibody. They actually discovered the original antibody we were using, Vanderbilt University, and they were able to actually use robotics to make many more antibodies than just one at a time. We now at Lawrence Livermore can also do that. We have stood up the same capability to make hundreds of antibodies at scale, to make and test very rapidly. So rather than just making one of these, we want to make over 300 of them. So they get arrayed in the way you're showing here. And in fact, what we do is we code robotics to do this for us. So what you see here is one of our biologists who's coded this up. He's watching it. And you see this robotic arm moving liquids from different tubes and different plates. This is doing the molecular biology. It's all programmed, it's computer programming, put onto this robot, and the robot is doing all this pipetting. So we don't have to do one at a time. And the robot does it exactly right. Don't make mistakes like the way a human might pipetting into a tube or a flask. Okay, this enables us to go super fast, be super accurate, and make tons of antibodies, because we don't know which ones are the ones that our computer simulations predicted to work will actually work. Okay, that's step 1, the DNA. A similar thing is true for step 2, where we take the DNA, put it in a cell, and we want the cell to make the protein, the antibody that we want to test. This is showing that similar thing. Computer programs are written, they are put onto this robot, and the robot does all this work moving things around to enable what I just described, where the cell will then produce the protein that we want, which is the antibody design. So there's 376 being made. And then the last step is to actually test that they actually work. Did one of our designs actually work to stop this new version of the virus, Omicron, from infecting human cells? Okay, so here is an example of our turns out to be our best antibody that we designed using what I just described. In blue, on this amino acid code, is what we changed. So you can see we made super precise, targeted changes that translate into changing the shape of the antibody to be just right to bind to that virus. We could never have guessed these changes. Remember, there's a quintillion possibilities for these changes. Much, much less likely than winning the lottery. But this was our best antibody, and we'll see how it worked. Here's a plot of the original antibody, which was fantastic against the original SARS, the original virus of the pandemic. And you can see it was 100% protective on the left. On the right, you see for Omicron, when SARS evolved, it made the antibody no longer bind. It also made it not protective because it didn't bind anymore. Our antibody worked for both. So huge success, high fives all around for the team. We got the antibody fixed, its shape was fixed, its chemistry was fixed so that it bound the new virus, Omicron, while also working for the previous versions of the virus. So now we have a repaired antibody that can now work and help save lives. Wonderful, wonderful thing. Never been done before, and we were super happy. We've now done this several more times for other viruses beyond just SARS. We've done it in cases where we took an antibody that works for one virus, and we made it work for multiple related viruses, so that a single drug can work for not just one virus, but several, which makes it much more effective and much more cost-effective for saving lives. We're continuing to do that. The next thing that we're doing, what we're working on now, actually, with several of our partners around the country, is what if we don't have a starting antibody? What if we don't have this scenario where there was an antibody that worked, and then it stopped working because the virus mutated, and we want to fix the antibody? What if we don't have any starting antibody at all? Now that problem, instead of having 18 zeros, instead of being a quintillion set of possibilities, it's way bigger. It's way bigger. It's like 100 zeros. Way, way more complicated because we have to come up with the entire code, right? Not just find out which changes work, but that entire code from scratch. Designing an antibody from thin air. That's what we're working on now, and that's called de novo antibody design, and that's the next generation of what I just talked about. That's what we're working on today. Okay, so let's do a quick review on what we learned. We learned how viruses can work to infect people, how they are super sneaky, super uncool, and they trick our cells into replicating themselves. So they get more and more virus in our body, making us sick. We also learned that antibodies can be equally sneaky, and they use a similar lock and key mechanism to bind to these viruses to prevent all that from happening. We also learned that those viruses can then try to escape that and change. And we learned that we can use AI to go back and fix that again and come back and restore protection to those viruses. Okay, this is the team that enabled all this to happen. This is the Lawrence Livermore team. We did work with a lot of partners, as I mentioned, Vanderbilt University, a lot of other partners that helped us with this. But this is our team at Lawrence Livermore. It's made up of software engineers, molecular biologists, AI experts, protein structure biologists, virologists. It's made up of optimization experts, all sorts of people coming together, different disciplines as a team all around this problem. That's what makes it work. And that's what makes it super fun and super impactful. Okay, thank you. I'll be hanging around for any questions.